Okay. Hi, everyone. Welcome to this webinar on the 16th of November. I feel a little bit, it was my birthday yesterday. I just want to let you know that, so I'm still celebrating a little bit, but I'm Anne-Catherine, as most of you know. And today we will be talking about mold in the webinar. And yeah, I'm very excited. It's been a while since we've done anything on mold. So yeah, I'm excited. So anyway, I'm Anne-Catherine, co-founder of Nordic Laboratories and DNA Life, and we are all about functional medicine and yeah, lifestyle changes, healing through nutrition and health optimization and yeah, all that stuff. So lab test, including this new report on mold, you can find in VMS, your practitioner profile. If you don't know how to find that, please reach out to us and we will support you so that you can get access to all the important tools that we are offering at Nordica and DNA Life. So I just want to let you all know that we are recording, and we will share the recording with you as soon as possible. And also the slides will be shared with you as well. As always, do put questions and answers if you feel like answering a question in the chat box. Remember, this is also about you feeling part of a community and getting to know each other a little bit more. And some of you, of course, may already have met each other at some of our live events around the globe. So the next webinar we have coming up on the 30th of November is on Grow Baby. We've had webinars on Grow Baby before, but this one is again with Emily Ritbom and Leslie Stones, Dr. Leslie Stones, on the Grow Baby test and on their research that they have done. And I have actually seen the presentation in September, where Dr. Stones presented at PLMI, and it was an honor to see Dr. Jeff Bland get up on stage and support her presentation, realizing that optimal health and prevention of disease starts preconception. So yeah, that was his endorsement of her presentation and the Grow Baby test. Today we have Dr. Teresa McBride with us, and she is a naturopathic doctor, a board-certified naturopathic doctor with more than 15 years of experience in both clinical settings and research and laboratory work. And today she's all into mold and toxic or mold toxicity in the urine test. So yeah, hold on to your chairs and notes and pens, etc., because I think you are up for a learning curve here. And do ask questions during the chat, and I'll present them at the end of the talk, and Dr. McBride will then help us answer your questions. So I will pass on the stage to Dr. McBride, and yeah, I'm looking forward to this.
Thank you. Hello, everyone. I'm going to share my screen and have some slides that will guide our conversation. I'm happy to finally connect with everyone. I hooked on an hour early. I was like, where is everybody? So the time change sometimes on Zoom gets messed up. And then the other thing that's going on here for me is the roofers are here, which I had no idea until this morning. So if you see me go like this or you hear a loud noise, it's something flying off the roof. So anyway, I wanted to let you know that before we start. So I'm going to share my screen and we will get going. So hopefully you can see my screen. Can someone verbally say that? Because now I can't see you guys because of my screen setup.
You can see your screen, or at least I can see the screen. So
Perfect. Thank you. Thank you. I just want to make sure it started sharing correctly. So kind of the main goal of our time together is to review the enhanced report to understand how it helps support your clinical interpretation and application of the results. We are also going to review some general clinical considerations when treating individuals with elevated mycotoxins. Talk a little bit about mold, but most of what we're going to are produced by molds and have very intense impacts on human health. And so we want to talk a little bit about what they are and why would you want to assess for exposure. And then again, we're going to review the new report features, which include a summary of elevated results. On the report, there's a human figure graphic and comprehensive test results. We also have a couple of ways for those of you who are familiar with the old test report format. There's been a couple changes in the way that the results are displayed, and so we want to go over those. There's also educational components of the new report design that provide an overview of the profile. There are detailed interpretations that address the individual mycotoxins. And then the report ends with a source chart. So how do you get exposed to mycotoxins via water damaged buildings and or food sources? And then we'll dive a little bit into the clinical approach to treating individuals who are experiencing adverse health effects from being exposed to mycotoxins. So what is the MycoTOX profile? So Mosaic's MycoTOX profile is a urine-based assay. So we're looking at urine and assessing levels of 11 different mycotoxins, including, and then the profile really helps you guide therapeutic strategy for prevention and treatment. So what are mycotoxins? They're secondary metabolites of certain mold species. So not all molds produce mycotoxins, but a lot of them do. And they're very pervasive. It's really becoming a global health threat. And they have numerous physiological impacts on health. And how mycotoxins impact any given individual really depends on a number of factors like age, sex, other medical conditions that they're dealing with, nutritional status, how long you were exposed to the molds that produce mycotoxins. So taking a closer look at the source and routes of exposure to mycotoxins, the main source of exposure are through water damaged indoor environments and food sources. So a lot of the food we eat is contaminated with mycotoxins. The routes of exposure, so how mycotoxins can enter the body. And by the way, this isn't just how you, this doesn't just address how you get exposed to mycotoxins, but also other toxins enter the body this way. You can inhale them, you can ingest them, again, through food you eat, and dermal contact from usually airborne spores. So those are the typical routes of exposure that we consider and look at. However, through clinical observation and the work we do with many clinicians, we're really revealing that there's, sort of another potential way that you can get exposed to mycotoxins, and that's through overgrowth in the GI tract. And this is often referred to as colonization in the gut. And we'll come back to this when we highlight our OAT test, organic acid testing, and mycotoxin as companion panels. And also want to mention, not shown on this slide, is the colonization of the sinuses. So often colonization not only happens in the gut, but we can also get colonization in the sinuses, and that's another potential source of exposure. So this idea of susceptibility to mycotoxins is really important, especially when you're dealing with members of the same household who might be responding differently. So the dose that you encounter from exposure to mycotoxins is dependent on the duration of exposure, the amount and concentration of what you were exposed to, the route of exposure. So inhalation, ingestion, or dermal contact have a different way that the body's going to absorb those mycotoxins, and then how frequently you were exposed. So all of that plays into the dose that you are exposed to, hence the toxicity of exposure to mycotoxins. And then this idea of individual variability. So again, everyone's going to respond differently, and that is dependent on things like underlying diseases the body's already dealing with, your genetics, your age, your gender, etc. So it's just a really thing to always keep in mind, because it's often challenging for the individual who is having an adverse effect from exposure versus the person in the same household who is not. So really explaining this concept and keeping it in mind is helpful when coaching patients. So when we consider the health impacts, a key point here is that if you do not address mycotoxins, other treatments tend to be less effective. So what we see clinically is that oftentimes these patients have other health concerns. For example, other co-infections like Lyme's or Bartonella or Babesia. So until you address the mycotoxins, the treatments for, say, Lyme's often is not as effective. But once you do address the mycotoxins, what we're seeing is the patient that wasn't responding to treatments previously, they now become more effective. So it's really seen more and more as a barrier or an obstacle to healing. And the way that mycotoxins impact the body is in a systemic way, leading to a wide range of toxicities that are shown here on this human figure. And they often present with a complex set of symptoms that vary from individual to individual, as we were just talking about. There's a difference in how any particular individual is going to respond to the mycotoxins. And they tend to be inflammatory. They often suppress the immune system, damage the mitochondria, and tax our ability to detoxify. And it's challenging for patients because mycotoxin assessment is often overlooked, especially if they're seeing, in the United States anyway, more conventional practitioners who often, or even functional medicine docs who are just not aware of how much mycotoxins can really be a barrier to healing. So I think that's the key takeaway on this slide. So when you're thinking about who would benefit from a mycotox profile, certainly what we just talked about is important. So, challenging patients suspected of co-infections is important. The other obvious consideration is anyone with an environmental history for current or past exposure to mold. So if in the intake process, this history of mold exposure comes up, that would certainly be someone you would want to assess for mycotoxins. And individuals who are exposed to mold spores can be exposed to mycotoxins, but they can also have just, the classic allergic reactions to mold and exhibit classic IgE type of symptoms, right? So your classic sneezing, sore throat, headaches, these kinds of things could be an IgE reaction. So if there is a known mold allergy, which, by the way, can be tested for, right? We have a test at Mosaic, the IgE molds allergy test. So if someone does have mold allergies, then certainly we might want to also consider assessing for the secondary metabolites of mold, mycotoxins. And the literature more and more is looking at associations with different diseases and mycotoxins. So there's at least some level of evidence in the literature for positive associations here. We're seeing associations with these diseases, and the list is growing all the time. Certainly when we talk about association, we can't say that they cause these diseases, but exposure to mycotoxins, more and more demonstrating positive associations with neurodegenerative diseases, in particular Alzheimer's and Parkinson's, impacts on gut health certainly are important, and then important to address these, as part of the treatment protocol. But seeing things like intestinal permeability, changes in the gut microbiome, as well as associations with IBD. And I think I left the citations in the slide, so you can dig a little deeper here. It's kind of interesting to look at how these associations are being made with mycotoxins. So that was just kind of, what are mycotoxins? Why would you want to test for exposure to mycotoxins? So let's jump into reviewing the enhanced test report. Over the next year or so, I imagine probably the next couple of years, Mosaic's going to be doing incremental enhancements to our test reports. So continually making small updates and enhancements. And these are really designed to improve the clinical value, right? We're really striving to do that, to provide more assistance with interpreting and applying the test results, and a real emphasis on education, not just for the practitioner, but also for the patient. So if they're looking at the report, they can get a better understanding of what mycotoxins are, why they're important to look at, and what that means for their health. So the first page of the report, we're going to walk through the pages of the report. And then there are things flying off the roof. Anyway, an important point to note is that this report enhancement is truly a report enhancement, and there are no changes to the reference ranges, no changes to the way the LCMS technology is measuring the actual mycotoxins. So that's just important to note. This first page of the report that's shown on the slide here provides right up front those mycotoxins with elevated findings. So previously we just listed out each mycotoxin, but here the report is pulling up to the front, again, those mycotoxins with elevated findings. This human figure graph should look familiar. We saw it on a previous slide. It's actually part of the report design. And elevated results are shown here within the context of this figure. And they're displayed in bold. So those same ones that were on that first page are displayed here again on the human figure. This it just helps to illustrate the potential widespread toxic effects of mycotoxins. Neurotoxicity, ox stress, immunotoxicity, hepatotoxicity, nephrotoxicity, GI toxicity, reproductive toxicity, and then looking at the carcinogenic effect of these mycotoxins. And then the next couple pages of the report, as we keep listing.
We just lost you a little bit there, like the internet connection just went out a little bit, so you might just have to go back a minute or so.
Do you want me to go back a couple slides? Is that what you're saying?
No, it's just like 1 minute or something, maybe that.
Okay. So should I go back one slide or just continue?
We lost you probably at the end when you were going through the previous slide.
Okay, this one. So I'm going to back up because I guess we lost internet connection there for a second. The next few pages of the report are simply showing the results for all of the mycotoxins measured. So that first page showed only the elevated. Here we're looking at the results for all of them. And then I want to take a little detour for those of you who are familiar with the previous version of the MycoTOX profile. There are a couple of changes to how the results are displayed. The old report, so this slide is showing the way we displayed results on the old report and the new report. So one change, which is shown here, is that findings below the detectable limit of the assay previously were reported out as a 0. On the new report, the enhanced report, they'll be displayed with a diamond that's placed at the far left of the results display, and it'll be noted with a less than DL, as shown here. There is a second way that there's a change to the way the results are displayed, and again, the previous result of the report is shown on top, and then the new version is shown below. Previously, when a result fell within the normal or green section of the report, we would just say less than the reference range. And on the new report, we're actually going to give you the exact numeric finding, and the diamond will show that numeric finding and be placed also on the green portion of the display bar. So a couple changes. They're actually more precise, but just so you're aware of them for those of you that are use the old report. And then continuing to go through the pages of the report, the remainder of the report is focused more on providing educational information about the report itself, and detail information about each mycotoxin. So this page is an overview of the report. It goes through what is the mycotoxin profile, a brief description, why would you care about testing for mycotoxins, a little bit about the testing platform, and a description of the reference range and how that relates to how the results are depicted on the report. So we're really finding that this page is, super helpful for patient education, helping them understand the importance of testing for mycotoxin exposure. Continuing to go through the report, it rolls into the interpretations for each mycotoxin. So these have been updated and thoroughly vetted based on a review of the literature. And here we're providing much more in-depth information about each mycotoxin, the source of exposure, the mechanisms of action, the impacts on health, and some helpful clinical insights. So this is provides a lot of good information. And what's not seen here, but via a link at the top of this page, you won't be able to see it, but it is there. If you go there, it will direct you to a ton of scientific literature in support of these interpretations. So if you want to dig a little deeper, that link will provide you access to literature reviews. And we've arrived at the last page of the report, or the last few pages of the report, which provide a chart. And it's condensing down some of the important information into chart format related to each mycotoxin. The chart is showing the sources of exposure from studies that actually measured mycotoxin levels. So whether that source is coming from water damaged buildings and or common food sources for each mycotoxin. We're also showing the genus and species of molds known to produce each mycotoxin, and then the potential toxicities from the exposure to those mycotoxins. So we've gone through the pages of the report. So now let's take a look at the clinical approach to treating the impacts from mycotoxin exposure. And I just want to talk about it, the general approach. And there's a lot of ways you can think about the clinical approach to treating mycotoxins. I like to break down the approach in this way. I think about it at 3 levels. And first, avoidance. So always with any environmental exposure, we want to take a detailed environmental history to try to understand exposures and follow up with patient education to support, removing the exposure. So that might be dealing with mold that's in the house, avoiding certain foods. If they've been exposed to other toxins, working with them again to remove and minimize the exposure. And then we want to support the foundations of health. So before we get into any more personalized or targeted therapies, we want to make sure that, in particular, that they're able to eliminate, right? So how's the liver, how's the kidney, how's the skin, how's the lung? So we don't want to start pushing toxins out of the body and they can't deal with it. You're going to make the situation worse. And then some simple things, but always helpful. And as mentioned earlier, a lot of times these individuals are really ill, and we want to make sure that they have adequate hydration. It's stunning how many people are dehydrated. And so really focusing in on that, because proper hydration also supports detoxification. And then we also want to assess their nutritional status. And so once we've done that, right, we get the person, we have an understanding of that, then we start considering more targeted, personalized therapies. So interventions that are going to help prevent absorbing more toxins and help with elimination of those toxins. So just stressing this point again, so removing the source of exposure, it's got to be removed or minimized. Otherwise, you're just going to keep getting exposed. So interventions focused on removal of mold. And often, this is in the home, but, unfortunately, we have to consider school, your office, your car, and again, food sources. A closer look at supporting foundations of health. So again, after we've identified and removed, hopefully, the source of exposure, the thing to think about before we start any targeted treatments focused on removing the mycotoxins is supporting these, again, foundations of health, making sure the pathways of elimination are optimized, what's going on with the person's organs of elimination, again, kidney, liver, skin, lung, other foundations of health. We talked about hydration, what's going on with the gut. So not only the liver, which is obviously a key organ of elimination, but the gut health in general, a lot of times is compromised from many things in our life, but mycotoxins can really have an impact on the gut. So we want to understand what's going on there. And then oftentimes folks with illnesses related to mycotoxin exposure, again, quite ill, and their nutritional status could use some support. So understanding their macro and micro nutrient needs is important. Common deficiencies that we see with these patients include vitamin D, magnesium, zinc, coenzyme Q10, B vitamin deficiencies, all of which, when you have deficiencies here, can adversely impact multiple pathways in the body necessary for detoxification. So again, just assessing what's going on there. These are, unfortunately, there's not one test, one diagnosis, and one treatment here. We have to kind of do the basics in terms of supporting health before we get into more, again, targeted treatments. So once we've supported these foundations of health, then we want to start considering personalized therapies that are more directed at preventing the absorption of the mycotoxins, helping to facilitate the removal. We don't want to recirculate the toxins, and any therapies directed at diminishing the toxic effects of exposure to mycotoxins. So typically, what's focused on first is binders. So how do we bind up the mycotoxins and get rid of them? And other considerations, sort of as the first line of strategy, is probiotics and glutathione. Probiotics help with the overall gut health, as we know, and much of the metabolism of toxins occurs at the intestinal biotransformation. So, and some probiotics are also help with binding and removing the toxins. And then we always want to think about glutathione. It can help bind toxins, but also in its reduced state functions on several enzyme systems in the body to assist in the detoxification, in particular of fat-soluble compounds, and as an antioxidant. And then usually thinking about adding on additional antioxidant support is important to consider here. So this is not an exhaustive list, but I think I put on the slide some antioxidants that there was some research behind that started looking at, particularly as it relates to mycotoxins. And then sometimes that's all that's needed, right? We assess the foundations of health, and then we work to bind and eliminate the mycotoxins. And sometimes additional things are needed. I kind of think of these as, quote, the big guns. So sometimes we need to disrupt the biofilm, which is holding the mycotoxins and other toxins, in particular heavy metals as well. And then often effective is the sort of, use of antifungals. And then there are a vast array of supportive therapies that we can look at that generally help with facilitating the removal. So things like exercise, induced sweating, some mycotoxins, not mycotoxins in particular, but toxins in general, are more efficiently eliminated through sweating. And then there are, a host of food and supplements that can also be directed at helping to bind, eliminate, and support detoxification. So this is a chart that I put together from 2 of our very popular blogs on our website, and the links are here on the slide. And this is based on research, which is somewhat sparse, but there is research out there which is looking at which binders work better against specific mycotoxins. So this is usually where targeted therapies start, and often do the trick without having to use, again, what I refer to as the big guns, biofilm disruptors and antifungals. So this can serve really as a roadmap when you're looking at the test results and then determining the binder treatments that you would want to consider. So I want to switch gears here a little and talk a bit about some supplements that practitioners are getting good results with. I'm not specifically endorsing any of these products. My intent is to let you know that these exist. A comprehensive list of all the supplements out there. But again, many practitioners that we work with are seeing positive results with some of these products. On this slide is Biocidin Protocol. I've seen this used frequently with what I would consider, patients who are in, I don't even know how to say it, good health, meaning, we've assessed the foundations of health and they're able to pull toxins out. And so you can just start with this protocol. And I encourage you to check in with these companies if you haven't. They have a lot of good teaching and research and support. But 3 products often used to address mycotoxins are these: Biocidin, GI Detox, and Proflora, which is a spore-based probiotic, which is all the rage right now as far as supporting diversity in the gut and dealing with issues related to intestinal permeability. This tends to be a 6-month protocol, 90 days suggested, really depends on the patient. But again, using things to support the gut, to break down the biofilm, to bind the mycotoxins, and support via probiotics, overall gut health. Here are a couple of products from Research Nutritionals. They have a nice product binder for effective elimination of mycotoxins, and they're also well known for their liposomal glutathione products. This is a product I think is relatively new. Actually, I don't know how long it's been out, but it's one that people are getting really good results with, addressing biofilms. So just some products to put on your radar if they aren't there already. Again, people seeing good results with, worth looking into. And all these companies provide education around specific treatments. So with the time that we have left, which isn't much, so I'm going to fly through these slides. Again, you'll get the slides, but I'd like to touch on assessing mold and mycotoxin illness by utilizing not only the mycotoxin profile, which we've been talking about, but also looking at organic acid, so our oat test. They really are best completed together if you have working with a patient with mycotoxin exposure. So like I said, they're really best completed together. It's convenient. You can run it on the same urine sample. And the two sort of main clinical insights that you gain is from looking at the oat in conjunction with mycotoxin is exposure. So more information about possible mold and mycotoxin exposure. And, as we know, the mycotoxin report looks for certain mycotoxins, and there are a lot of mycotoxins and molds that produce mycotoxins that we don't measure on that report, but that we can gain some insight to when looking at the oat. And then the other important thing that running an oat along with mycotoxin provides is the impact. So it really helps support assessing the systemic impacts that mycotoxins can have. So mitochondrial function, how well is someone detoxifying, what are the nutritional deficiencies? We talked about that previously, that a lot of these patients are depleted. So what are their nutritional deficiencies? And oxalates is an important thing to look at. They can be formed from candida and mold and have an additional impact on mitochondrial function. So briefly diving into the oat real quick, this can get overwhelming pretty quickly. But as far as widening the lens on the possible exposure, this first page of the oat is really helpful. So we can look at fungal activity, this idea of colonization in the gut, and this being another source of mycotoxins. So markers on this first page, 2, 4, 5, 6, can be indications of Aspergillus, 9, Fusarium. So this can be from a recent or a past exposure. Also important to look at Candida with these patients. The key indicator of this is elevations in arabinose. We often see this elevated with mycotoxins. Gliotoxin, one of the mycotoxins measured on the MycoTOX profile, is suspected to produce candida. So we've got this connection with candida and mycotoxins going on that we can see, gain more insight into by looking at the oat. Also, Clostridia bacterial markers on this first page are often elevated with mycotoxins because they're opportunistic, right? In and of themselves, they provide some insight into the microbial environment of the gut. And they also tie to other sections on the oat, mainly neurotransmitters, which is a little beyond the scope of our talk today. But and then there are other organic acids that indicate mold and mycotoxin exposure throughout the report, but this page really is where the insight is, and would certainly be where, if you're newer to utilizing organic acids, this would be where you would focus to take a closer look at potential exposures. And then there's a whole host of organic acids that help us understand mitochondrial function. So the oat provides a look at the organic acids involved in the Krebs cycle. So along with the electron transport chain within the mitochondria, these are the main pathways that produce our energy. So as you probably know, the mitochondria is often referred to as the powerhouse of the cell. So macronutrients that we eat, protein, lipids, carbs, all feed into this cycle to produce our energy. So again, a lot of markers throughout this test that help us see how the mitochondria is functioning, but this set of organic acids really provide insight into needed supportive therapies, which might include CoQ10 or L-carnitine, etc. So the oat additionally gives some insight into metabolic byproducts associated with mold and mycotoxin exposure, and one key one here is oxalate. So these crystals increase in cases of mold exposure, and good research that linked species of Aspergillus and Penicillium to producing oxalates. And another tie into Candida can influence oxalate production as well. Here we can look at nutritional status. We've already talked about how there's sort of generalized nutritional deficiencies with these patients often. Things we see is B12, need for B12, B2, again, CoQ10. These are just some. So trying to wrap it up here, want to remind you guys of some important resources that will help with understanding mycotoxins, and these are available on our website. These are some of the popular blogs, particularly helpful in addressing therapeutics. So there's a series related to the use of different binders. That previous slide, I had created that chart. That was from a couple of blogs, popular blogs that we have addressing the use of binders. We also have a ton of webinars focusing on in-depth toxic topics related to assessing and treating mycotoxin exposure. And they really dig into the dangers and health impacts and what you do about them. So a lot of great webinars. I encourage you to check them out. And then we also have Mosaic Edge. So we have free monthly webinar series. We always try to teach organic acids fundamental seminars. And then we have Edge workshops that dive deeper into particular topics. And those happen quarterly, I believe. And last but not least, we have, we have a team of clinical educators on our team, and just highly recommend utilizing this complimentary service, especially if you're new to any particular report. It's helpful to get on the phone with someone, review the case, and kind of work together to interpret the results and you know, how you would apply them to the particular patient you're working with. So, wow, I ended exactly at 10 minutes left for questions. That's pretty good.
Thank you very
I'm gonna
much.
stop sharing.
So I took notes of some questions that came up.
Okay.
And I'll just throw them out as they come. But the first one is that if you check for creatinine in the urine and the concentration is too low, do you ask for a new collection, or do you just do the test?
Well, we check it at the lab. So if it's too low, we we note that and reach out and let you know. If you notice it's low and you have concerns, don't hesitate to reach out to us, and we can double check that, make sure that is not impacting the results. So we do double check it before it goes out, though. If it's too low, we won't report the results out.
Okay. So one asked, because I know there are some tips in order for that you can mobilize more toxins before you do the collection, so that you get a more positive result, or perhaps more precise in regards to. Do you have any tips on how to mobilize toxins before you collect the urine?
So this is an area which we need more information on. So our urine assay is LCMS based, and so we're looking at unconjugated free mycotoxins. So when you bind things, you're typically conjugating them. So, you know, the effect that something like glutathione, we recommend not doing that before our test, because you're going to bind it and it won't be read via LCMS. So other methods used in urine analysis look at both bound and unbound. So both methods are accurate at assessing mycotoxins in urine. That's just the difference. With glutathione, it's tricky because we don't really know the impact. And that's why we say to avoid it before taking the mycotox profile. It does have the ability to bind mycotoxins, but it has a lot of other things that it does, right? It just helps overall elimination in general. So because it has so many effects on the body, we just really don't know the impact. So in our particular test, just to be safe, we say, avoid using glutathione prior to taking the test. Sorry, it's kind of a complicated arena. Hopefully that helped a little bit.
That's okay. And then, so then a question came up, which was actually before the webinar, a person wrote to me. So if it's a person who's like working in a mold exposed building and they do a test and it comes out more or less negative, could that be because they are binding it and not excreting it, or poor detox capacity? Or how should we interpret if we know that the patient is exposed to mold, sample comes back negative?
Yeah, there's a few things that could be going on there. One, I think you mentioned that their ability to detoxify is typically what's going on. And then you would want to look at what's the clinical picture. So again, susceptibility individual to individual varies. So someone could be exposed, it's not affecting them. But if you have a patient that's exhibiting some symptoms and you suspect it's from exposure to mycotoxins, then yeah, you would be concerned about their ability to detoxify those mycotoxins, and that's why you're not seeing them. Also, again, we look for the most common mycotoxins, but it could be they are eliminating mycotoxins that we're not measuring for.
Good point. So when you say antifungals in the treatment protocol, you mean both natural and prescriptive antifungals like diflucan, etc.?
I am, and that's very much dependent on the practitioner and their prescriptive rights, their comfortable, their comfort level with that. This is a whole area of different viewpoints, right? A lot of practitioners have found using antifungals to be very effective, especially with patients who you've done a lot of other things to try to address the mycotoxins and they're still having issues. You take that step and it really helps. Other people see that as, well, you're disrupting the gut microbiome with antifungals, and what are you treating exactly? So it's an area to really it's in need of deeper exploration, and people have found it very valuable. And yes, that can be prescriptive, and then there's a whole host of herbal antifungals that people have found positive results with. So yes, both prescriptive and natural. And also the point I want to make there too is a thing to maybe attempt before antifungals is biofilm disruption. So you maybe take that next step because they're bound up in the biofilm. You're using a good set of binders. That works. You don't need to go to antifungals. Again, that's why I call that a big gun. So getting those other things, trying those other things first, but that's a clinical judgment call.
a few questions that comes up as well is the exposure, like previous exposure or current exposure. So like if you have the OAT report, and for that matter, the mycotoxic report, do we know if it's now or if it has been, and if they have been removed from the source?
Well, knowing if it's current or not relies heavily on your environmental history intake, right? So through talking and understanding where they live and where they work and all those things help to know what they're currently being exposed to. But the oat can help, again, looking at the gut and the possible colonization there. That colonization can be a result of an exposure that happened in the past. Also, I think I mentioned, but wasn't on the slide, nasal colonization too. We can think of those either being from something you're currently continuing to be exposed to, or it's from a past exposure.
So then another question came up that apparently a doctor had advised not to do the protocol because it would disrupt the medication, because the patient was on thyroid medication. But surely you just time the intake of the medication and the biofilm disruptor or the binder, etc., like we would do in all other protocols. So maybe it's just the doctor that is my question. Like maybe it's just that doesn't
yeah, I mean, there is some consideration on timing. You mentioned it. Because the binders can bind medications, they can bind other things too, depending on what you're using. So that's something to consider in your dosage strategy and your timing.
Okay, well, I think your time is up. So thank you for your time. And yeah, I look forward to get the slides for you, because I now share both the recording and the slides with everyone. So yeah, thank you very much.
I enjoyed it. Appreciate all of your kind attention, and hopefully you didn't hear the stuff flying off the roof. I was like, I feel like I'm like in a war zone here. It was like, they're shooting off the roof. The roofers are here. I told you guys. But anyway, so I hope this was helpful. And yeah, I will get the slides to you. So yeah, again, many thanks, you guys.
Enjoy your day.
Thank you. You too.
Bye.
Bye.